present our paper, Evaluation of Correctness in Unsupervised Many-to-Many Image Translation by Dina Bashkirova, Ben Usman, and Kate Sayenka. Given a pair of semantically similar but visually different domains, unsupervised image-to-image translation aims to learn a mapping of images from one domain to another so that the translation result is a plausible example from the target domain and the content of the input image is preserved in the translation result. One of the most significant applications of image-to-image translation is the fundamental problem of domain adaptation. We're given a labeled domain of images, we learn a mapping to the unlabeled target domain to generalize to new unlabeled data. However, the one-to-one formulation is ill-posed since it assumes a one-to-one correspondence between the images in two domains, which is not the case in the majority of applications. Therefore, we consider a many-to-many approach where we are given an additional guidance image from the target domain that specifies the domain-specific information of the translation result. For example, whether or not the translated image should have a beard in the female-to-male translation case. The popular evaluation metrics for image-to-image translation are the FID and inception scores that measure realism, the perceptual similarity score that evaluates the diversity of outputs, and in rare cases, human evaluation that also measures realism. The problem is that none of these metrics evaluate the semantic correctness of the translation result, which is crucial to make image translation more reliable and applicable in the real world. A correct translation must preserve the factors that are shared across domains, same as in the input image, while manipulating the domain-specific factors according to the guidance image. A picture on the right illustrates an example of a highly realistic but semantically incorrect translation. The hair color is the shared factor which should have been preserved, while the facial hairstyle is the domain-specific factor and therefore it should be taken from the guidance image. We propose an evaluation pipeline based on the data sets with labeled factors of variation. We construct the two domains by choosing the non-overlapping sets of domain-specific and shared attributes and filter the data accordingly. This way, we obtain a pair of domains with known shared and domain-specific factors by design. The evaluation for a given pair of input and guidance images consists of comparing the attributes of its translation result with the attributes of a semantically correct translation. This way, we constructed three datasets: the Cell-A-Day male and female dataset, the 3D shapes dataset, and the Synaction dataset. We also developed a set of metrics measuring the semantic translation quality, content preservation quality, and domain-specific manipulation quality. We report the quantitative evaluation results in our paper. Here we summarize the key insights of our experiments. First, there is a clear trade-off between the content preservation quality and domain-specific manipulation quality. Typically, the failing methods fall into one of two categories: the ones that preserve content well, but fail to translate the image correctly or manipulate the domain-specific attributes, and those that perform the translation and manipulation well, but fail to preserve the shared content information of the input image. Second, all methods we tested fail to add or remove certain objects, such as facial hair. Third, methods that use adaptive instance normalization, or ADA-IN, have an inductive bias towards treating the color and texture-based information as domain-specific factors, regardless of whether or not it's actually the case for the given dataset. For example, they change the hair color better on average than the non-ADA-IN based methods in Cell-A-Day experiments. They also change the object color in the 3D shapes experiments, even though it's supposed to be preserved. The non-ADA-IN based methods manipulate the colors and textures poorly compared to ADA-IN based methods. Lastly, all methods we tested preserve the overall scene layout and poses correctly on Cell-A-Day and Synaction experiments. Thank you very much for your time, and please check out our project page.